My name is Ron Q. I'm a professor in the crop science department working on crop biotechnology. Today's lecture is about genetic transformation of plants. The content is condensed from a lecture for a graduate student course. Genetic transformation of plants is also known as plant genetic engineering or plant biotechnology. In this approach, we introduce new genes into plants. So the plants are called transgenic plants or sometimes GM crops, that is, genetically modified crops. The approach is also called molecular breeding since it is an extension of the conventional breeding for plant improvement, but using molecular techniques. The specific contribution of molecular breeding is that it can break the crossing barriers between genera, families, and even kingdoms. For example, genes from bacteria have been introduced into plants to confer resistance to herbicides or insects. The advantage of molecular breeding over introgression used in conventional breeding is that it can introduce a single or a few stacked desirable genes without introduction of any undesirable genes. The first transgenic plants were obtained in the early 80s. GM crops were first released to the farmers in 1996. They were grown on 4.3 million acres that year, and their acreage has steadily increased ever since. From the figure you can see, the global acreage of GM crops in 2010 reached 366 million acres, 90-fold higher than that in 1996, and is about 10% of the cultivated cropland in the whole world. The GM crops are mainly used for production of soybean, maize, cotton, and canola. In soybean production, 81% of the global acreage is already transgenic. The major traits of the first generation of the GM crops are herbicide tolerance, insect resistance, and the stacking of these two traits. Why are GM crops so popular? It is because it reduces production cost for farmers and increases yield and thus farmers' income. In the first 14 years, the global economic gains are $65 billion. The transgenic traits are stable and no major breakdown has been reported since its first commercial release. Moreover, transgenic food and feed seem to be as safe as non-transgenic, and no safety-related incident has been reported in the past 15 years. By now you may wonder how we can introduce a gene into plants and make it work. First, it involves a technique called plant tissue culture. That is, place certain organs or tissues of a plant onto a culture medium under aseptic conditions. Quite often, a cluster of undifferentiated cells called callus would be induced. When changing plant hormones in the medium, we often can recover new plants from the callus. If we introduce genes into plant cells at the callus stage, we will be able to obtain some plants that have the introduced genes, and this would be the transgenic plants. How can we make an introduced gene work in plants, or in our term, to be expressed? To make a gene expressed properly, we always need to have a suitable gene promoter and a terminator. Promoter is a gene switch to determine when and where the gene is expressed and how much it is expressed. The terminator ensures the gene transcription stops at the right place. When we put a promoter, a gene, and a terminator together, it is called a gene construct. Usually we use restriction endonucleases and DNA ligases to make gene constructs. We then multiply the gene construct in a small circular DNA called plasmid in E. coli. After the gene construct is made, we need to deliver the gene construct to plant cells. The most widely used method utilizes a bacterium called Agrobacterium. It has a natural ability to insert a piece of DNA fragment into plant genome during infection of plant cells. The second method involves a so-called gene gun to deliver DNA into plant cells by bombarding plant cells with tiny bullets, with a gene construct on the surface of the bullets. The third method also uses Agrobacterium, but does not have to go through tissue culture at all. We only need to dip the flowers in Agrobacterium suspensions. It is called floral dip. Agrobacterium tumefaciens is a soil-borne bacterium, and it causes crown gall disease. Naturally, Agrobacterium is capable to insert a defined piece of DNA called tDNA or transfer DNA from its own plasmid called Ti plasmid into plant genome during infection. The tDNA contains some genes that cause tumor. So these genes are called oncogenes. By removing these oncogenes, the Ti plasmid is called disarmed and will no longer cause tumor formation. So we can insert our gene construct at the tDNA region for plant transformation. On Ti plasmid, there is a cluster of virulence genes which are responsible for the infection and tDNA transfer. It was later found out that the virulence genes work in trans mode and do not need to be on the same plasmid with a tDNA. So the binary vector was created with the tDNA region on it. Now the Agrobacterium has two plasmids in it. One has virulence genes but no tDNA, called helper plasmid, and the other binary vector with tDNA but no virulence gene region. The binary vector is much smaller and much easier to manipulate. The gene gun method is also called the ballistic method, particle bombardment, or microprojectile bombardment. The gene gun has a few versions, and the Bio-Rad PDS 1000, as shown here, is the most widely used one. We coat the plasmid DNA containing the gene construct on the tiny gold or tungsten particles, about 1 micrometer in diameter, and use high-pressure helium gas to accelerate them so they can penetrate the cell walls. The DNA has to enter the nucleus for expression. The blue color marked cells in the lower right figure shows the cells in a piece of callus where the transgene is expressed after bombardment. Floral dip directly dips the young inflorescences into agrobacterium suspension to deliver gene to the ovules, so to yield transgenic seeds. By doing so, it circumvents the tedious tissue culture procedure. However, it has a narrow developmental window to work, and it only works in a couple of species, including the model plant Arabidopsis. The transformation efficiency is about 1 to 3%, as shown in the bottom figure, where only transgenic seeds can germinate. In transformation experiments, we often want to get an early feedback of our treatments and to see where our gene goes and expresses. It is not easy with a regular gene since it is hard to detect. That is why we need so-called reporter genes. There are several reporter genes available, and we will focus on 2 such genes that are mostly used in plant transformation. The first one is called the Gus gene from E. coli. Gus gene encodes beta-glucuronidase, an enzyme that converts a soluble, colorless substrate called X-gluc to insoluble indigo to give a blue color. After a transformation experiment with Gus gene, we can feed the callus with X-gluc and observe the blue color spots for the Gus gene expression, as shown in the upper panel. In a couple of months, we may see a whole piece of callus becomes blue, as shown in the lower panel. The Gus enzyme activity can be quantified using another substrate called mug by fluorometry. Another reporter gene is called green fluorescent protein, or GFP. GFP is a protein found from jellyfish. The advantage of GFP over Gus is that it is non-destructive. You can view the callus in culture under a fluorescence microscope. The disadvantage is that GFP expression is not easy to quantify. Here shows GFP expression in cells and some plant tissues. Using the reporter genes, we can conveniently detect transgene expression in plant cells. There are 2 kinds of transgene expression. One is called transient expression, as shown in the upper panel. That is when the gene construct first enters the nucleus, the transgene could be expressed. But if the transgene is not integrated into the plant genome, the DNA will not stay long and eventually be degraded and no longer be expressed. In this case, the expression is short-lived and thus transient. If the transgene is integrated into the plant genome, it will be inherited to the daughter cells and will have a good chance to be expressed in all the daughter cells following cell divisions. In that case, we would see expression in a cluster of cells and eventually in all the tissues of a transgenic plant, as shown in the lower panels. This is called stable expression. In general, only a small percentage of transient expression events become stable expression events. To compare with the total number of cells we use for transformation experiments, the stably transformed cells are a tiny fraction of it, and would be very difficult to identify and separate. An effective way to isolate the stably transformed cells is to use a selection system. The system includes a toxic chemical in the culture medium and the introduction of a selectable marker gene into plant cells that can detoxify the chemical effect. One example is selection with an antibiotic, such as kanamycin or hygromycin B. Both suppress plant cell growth by interfering protein synthesis. The encoded enzyme by the selectable marker gene MPT2 or HPT, respectively, can chemically modify the antibiotic and detoxify it. So only the plant cells that have the selectable marker gene can grow in the selection medium containing the antibiotic. The figures show the selection effect of hygromycin B on callus and plants. Some herbicides can also be used for transgenic selection. An example is a herbicide PPT, also known as phosphoenoltrypsin or glufosinate. PPT is an analog of glutamic acid, binds and inhibits glutamine synthetase, and causes ammonium accumulation to kill the cells. A related herbicide, biolaphos, is a tripeptide containing PPT. The resistant gene, called BAR gene or PAT, acetylates PPT so it would no longer bind to glutamine synthetase and thus becomes non-toxic. So PPT in the culture medium and the BAR gene in transgenic cells form a selection system. A similar herbicide selection system is the Roundup herbicide and the CP4 EPSP synthase gene. Unlike the antibiotic selection systems, the herbicide selection makes the transgenic plants resistant to a herbicide, which is a valuable agronomic trait by itself. The photos here show transgenic selection using biolaphos. After obtaining resistant plants from the selection, they need to be analyzed and characterized at molecular level to verify they are transgenic and to demonstrate transgene expression. Southern blot that analyzes genomic DNA can be used to demonstrate the presence and integration of the transgene, to distinguish different transformation events, and to estimate transgene copy numbers. PCR can sometimes be used as a quick and preliminary method to evaluate transformation, but not as reliable as Southern blot due to possible DNA contamination, especially from Agrobacterium. Northern blot, as shown here, reverse transcription PCR, Western blot, and ELISA are often used to evaluate transgene expression. In addition, we need to see whether the useful transgene we introduced into plants work as we expect. For herbicide resistance gene, we can apply the herbicide to the transgenic plants to see whether the transgene makes the plants resistant to the herbicide. For other transgenes, for instance in this case from Dr. Hong Luo's lab at Clemson University, they introduced a salt tolerance gene into plants. They need to evaluate the plants under salt treatments to see whether the transgenic plants are more salt tolerant than the non-transgenic plants. They analyzed the leaf, fresh and dry weight, chlorophyll content, root development, and other parameters when treating plants with various salt concentrations. It is best if we can establish a correlation between plant performance and the transgene expression level to clearly demonstrate the transgene effects. You may have a question regarding how the transgene is inserted and integrated into a chromosome. We know very little about it, but by analyzing the insertion sites through bombardment or agrobacterium-mediated transformation, people believe that the insertion is mostly random and the inserted sequence has little homology with the host DNA sequence at the insertion site. And this is called illegitimate recombination. It is observed that transgene expression level varies among transgenic plants, and believed it is caused by where the transgene is inserted. This phenomenon is called position effect. Quite often, if the plants have more than one transgene copy, they inherit together as one locus, no matter whether it is obtained by agrobacterium transformation or by bombardment. Sometimes silencing of transgene expression is observed, which is usually associated with multiple transgene copies. Finally, we need to demonstrate transgene inheritance to the offspring. As shown in the diagram, the transgene is generally inherited in a Mendelian manner, which is 3:1 ratio. Sometimes 1:1 ratio is observed, and the reason is not clear. By now we only talked about nuclear transformation, that is, the transgene is inserted into a chromosome. Actually, chloroplast can be transformed too. In contrast to nuclear transformation, insertion of transgene into chloroplast DNA is achieved through homologous recombination by design. Thus, there's no position effect in chloroplast transformation. Moreover, since each cell has many chloroplast DNA copies, and there's no gene silencing mechanism within chloroplast, transgene expression could be very high. So chloroplast transformation can be used to maximize accumulation of a desirable protein. Chloroplast transformation is also considered a way to prevent transgene escape through pollen. However, the limitation is, so far, chloroplast transformation has been achieved in only several species. Well, this concludes the lecture. Thank you for viewing. Goodbye.